Today we shall study packing of DNA helix. It is for 5 marks. Explain packing of DNA helix. We know that DNA helix is very long, highly coiled and tightly packed in the nucleus. The human DNA in a cell contains about 6.6 multiplied by 10 to the power of 9 base pairs. This approximately comes to 2.24 meters of DNA. That is, the human DNA length in a cell is 2.24 meters. A neat point. It is more than dimension of the nucleus. That is, if we compare the length of the DNA and the dimension of nucleus, the DNA length is more. Then, how to calculate the length of DNA? This we have already seen in previous class. The length of DNA is calculated by multiplying the total number of base pairs with the distance between two base pairs. For example, in man, the length of the DNA can be calculated like this: 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of minus 9 base pairs. 10 to the power of minus 9 and 10 to the power of plus 9 cancelled. Then 6.6 into 0.34, that comes around 2.24. So this is how the length of the DNA can be calculated. Now, packing of DNA. How this packing DNA is packed in prokaryotes and the DNA is packed in the nucleus of eukaryotes. First, we shall study the prokaryotes. You know, prokaryotes do not have definite nucleus. The DNA is found in the cytoplasm of the cell. In prokaryotes, that is example, bacteria, the negatively charged DNA held with some positively charged proteins and form a coiled structure, a loop-like structure called nucleoid. So in prokaryotes, the nucleus is also called nucleoid. It's also called genophore. It is circular. That is, DNA is circuloid. So this is how the DNA is packed in prokaryotes. Now we shall study how the DNA is packed in eukaryotic nucleus or in eukaryotic cell. In eukaryotes, the negatively charged DNA wrapped around positively charged protein octamere, that is histones, form a structure called nucleosome. In eukaryotes, DNA is stabilized with positively charged basic proteins. Positively charged proteins are the basic proteins. Basic proteins means histones. Histones are the basic proteins. So these histones are the basic proteins are rich in amino acids like lysines and arginines. It's a very important, neat question. Now, the basic histones are present in octamere. So, there are four types of basic proteins. Example, H2A, H2B, H3 and H4. These are present in octamere. That is, histones are organized to form 8 molecules called histone octamer. You can see the diagram. 8 second diagram. You can see 8 histones. In the first diagram, you can see 4 histones. That is H2A, H2B, H3, H4. ಡಯಾಗ್ರಾಮ್ನಲ್ಲಿ ತುಂಬ ಚೆನ್ನಾಗಿ ಕಾಣುತ್ತೆ. DNA, I told you, is wrapped around the protein. Sometimes we call protein core. This DNA helix contains 200 base pairs. It's very important, neat point. Nucleosomes are closely packed, form a thread-like structure called chromatin fiber. If we examine this under the electron microscope, the chromatin fiber appear like beads on a string. ಇದು ಒಂದು ಮಣಿ ಸರದ ಥರ ಕಾಣ್ತಾ ಇರ್ತದೆ. Nucleosomes form the repeating unit of a structure. This chromatin is packed. So nucleosome is packed and form chromatin fiber. I told you. This chromatin fiber further coiled and condensed. Condensed means become short during metaphase stage of cell division and form a structure called chromosome. This is how the chromosomes are formed. So nucleosomes are the fundamental structures. You can refer the metaphase chromosome diagram also. Next, another important point is also there. Now, the chromatin fiber coiled and condensed to form chromosome. What helps in condensing means, so the packing of fiber takes place by non-histone chromosomal proteins. That is NHC, non-histone chromosomal proteins. Very important point, I told you the proteins of nucleosome are made up of histones, but the packing proteins are non-histone proteins. A very important notable point. So that's all about packing of DNA helix. Now we shall study the search for genetic material. Transforming principle, that is Griffith transformation experiment. Actually, we are going back. So far we have studied the modern structure of DNA and the fundamental nature of the DNA or the chromosome. That is, chromosomes are made up of fundamental units, nucleosomes. The structure of DNA was discovered in 1953. You know that Watson and Crick discovered the structure of DNA. Now actually we are going back. ಹಿಂದಕ್ಕೆ ಹೋಗ್ತಾ ಇದ್ವಿ ನಾವೀಗ ಮಾಡರ್ನ್ ಬಿಟ್ಟು ಹಿಂದಕ್ಕೆ ಹೋಗ್ತಾ ಇದ್ವಿ 1926ಗೆ ಹೋಗ್ತಾ ಇದ್ವಿ ನಾವು. So look here now. The discovery of Maclyn, it was discovered in the year 1869. The nucleic acids were discovered by Frederich Griffith in 1869. And loss of heredity was discovered by Mendel, Gregor John Mendel. Both almost at the same time. But we have taken more time to prove DNA is the genetic material in the living organisms. This is the introduction. Now we shall study the actual experiment. In 1928, Frederich Griffith discovered the process of transformation in a bacterium called Streptococcus pneumoniae. This bacterium, formerly known as Diplococcus pneumoniae, it causes pneumonia in man. Pneumonia means a type of respiratory disease, not only in man and also in some other mammals. This bacterium occur in two strains, smooth and rough strain. Smooth strain. The smooth strain bacterium. When this type of bacterium grown in the laboratory, the bacterial colonies appear smooth and shiny. It, the shining nature is because of outer coat or the capsule, a polysaccharide. So this type is called S strain. It is a virulent strain or pathogenic in nature. R strain. This type of bacterium, when grown in the lab, produced rough colonies. So in this bacterium, capsule was absent. R strain actually non-pathogenic. If you refer some other neat books, you may come across the S strain further classified into S1, S2, S3 and R strain into R1, R2, R3. Now the experiment we shall study. You refer the diagram also, easy. Now there are 4 or 5 points. If you write these 5 points, you will get 5 marks. Now, experiments. In one of the experiment, when the R strain bacterium are injected into the mouse, it is non-pathogenic and does not cause pneumonia. The mouse was alive. In the second experiment, when the S strains are injected into the mouse, that cause pneumonia and mouse dies. He conducted another experiment. He killed S strain bacterium by heating. That is, when heat killed S strain bacterium injected into the mouse, that does not cause pneumonia. The mouse was alive. He conducted another experiment. Heat killed S strain bacterium and mixed with living R strain bacterium injected into the mouse. The mouse dies because of pneumonia. Conclusion of the experiment. R strain bacteria have been transformed into S-strain bacterium. That is, the transformation of R strain into S strain is because of transfer of some genetic material. So he concluded that R strain is transformed into S strain because of some transforming principle. Some transforming principle. The actual biochemical nature of the transforming principle was not known at that time. At that time means the Griffith time. So that's all about Griffith experiment. It is very important experiment for 5 marks. Explain Griffith transforming principle. Question. Now we shall proceed to the next experiment. So let us recapitulate. In Griffith experiment, R strain transformed into S strain because of some transforming principle. What was actually the transforming principle? That was not known. To discover, further experiments conducted. So now the heading, Biochemical characterization of transforming principle. Experiment conducted by O. Avery, C. Macleod, and M. McCarty. That is, Avery, Macleod, and McCarty conducted again series of experiment in 1932 to 44 and worked to determine the biochemical nature of transforming principle of Griffith experiment. They actually purified the biochemicals from heat-killed S-strain bacterium. Biochemicals means the bacterial cell contains proteins, DNA, and RNA. These biochemicals are purified. Then they added digestive enzymes of each. Digestive enzymes means protease enzyme to digest proteins, RNase enzyme to digest RNA, DNase enzyme to digest DNA, to see which one could transform R cells to S cells. Now see the diagram. Heat-killed S strain mixed with protease enzyme and R strain transforms R strain to S strain. This is one of the experiment. Once again, heat-killed S strain plus protease enzyme plus living R strain. So transforms R strain to S strain. He conducted another experiment. He killed S strain mixed with RNAs. RNAs means, RNA digesting enzyme and R strain. After this experiment, he found R strain transformed into S strain. Then he conducted another experiment. He killed S strain plus DNase enzyme taken in the medium and R strain. Then he found R strain does not transform into S strain. So this is how he conducted experiment. By this experiment, we can come to a conclusion. When we added protease enzyme, protein digested. When we added RNase enzyme, RNA digested. So no transformation was found. Hence, we can come to conclusion that protein and RNA is not the genetic material. But in the experiment, DNA is added, the transformation was not found. So it concludes that DNA is responsible for the transformation. So what was the transforming principle according to this experiment? The DNA. But all biologists at that time are not convinced about this experiment. Now, one beautiful question is there, that is differentiate DNAs with DNAs. ಇಲ್ಲಿ ಸ್ಪೆಲ್ಲಿಂಗ್ ಅಷ್ಟೇ ಮಿಸ್ಟೇಕ್ ಇದ್ದಂಗೆ ಕಾಣುತ್ತೆ. ಇದು ಕೊಟ್ಟರೆ ತುಂಬ ಕಷ್ಟ ಬರೆಯೋದು. ನೋಡಿ ಇಲ್ಲಿ DNA apostrophe S means different types of DNAs. For example, bDNA, cDNA, zDNA, like that. DNASE means it is an DNA digesting enzyme. This is how you have to differentiate. So that's all about this experiment, biochemical characterization of transforming principle. Next, further experiments conducted by different scientists to prove the DNA is the genetic material. In Griffith experiment, some transforming principle responsible for transformation of R strain into S strain, that we came to know. In biochemical characterization experiments, we came to know that transforming principle was DNA. Now we are conducting the experiment to prove that DNA is the genetic material in living organisms. This experiment is called Hershey and Chase experiment. It's also for 5 marks. Now the heading, the genetic material is DNA. DNA is the genetic material, is proved by Hershey and Chase in the year 1952. Who proved DNA is the genetic material? Hershey and Chase. They conducted experiments in bacteria using the virus. The virus that infect bacteria called bacteriophage. What is bacteriophage? The virus that infect bacteria are called bacteriophages. Now we shall see the experiment. During infection, the bacteriophage first attaches to the bacterial cell wall. This you have studied in your first PUC. Then it injects or inserts its genetic material into bacterial cell. ಫಸ್ಟ್ ಸ್ಟೆಪ್ನಲ್ಲಿ ನಿಮಗೆ ಗೊತ್ತಿದೆ, ವೈರಸ್ ಇನ್ಫೆಕ್ಟ್ ಆದಾಗ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ ಒಳಗಡೆಗೆ ವೈರಸ್ನ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ಲು ಎಂಟರ್ ಆಗುತ್ತೆ. ಡಯಾಗ್ರಾಮ್ ಇದೆ, ರೆಫರ್ ಮಾಡಿ. Next, the viral genetic material become part of the bacterial genome and it starts manufacturing virus particle using the DNA of the host. Host means bacterium. So this knowledge is very important. This is the fundamental knowledge, or you can say how virus reproduce in a living cell. ಈಗ ನಾವೇನು ಹೇಳ್ತೀವಿ, ಆ COVID-19 ನಮ್ಮ ಸೆಲ್ಲಲ್ಲಿ ಬಂದರೆ ಹೆಂಗೆ ರೀಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ ಅಂದರೆ ಇದೇ ಥರ ರೀಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacterium. Hershey Chaseಗೆ DNA ಅಥವಾ ಪ್ರೋಟೀನೇ ಜೆನೆಟಿಕ್ ಮಟೀರಿಯಲ್ ಆಗಿ ವರ್ಕ್ ಮಾಡೋದು ಅಂತ ಕಂಡುಹಿಡಿಬೇಕಾಗಿತ್ತು. ಅವರು ಎಕ್ಸ್ಪರಿಮೆಂಟು ಕಂಡಕ್ಟ್ ಮಾಡ್ತಾರೆ. ಸೀರೀಸ್ ಆಫ್ ಎಕ್ಸ್ಪರಿಮೆಂಟ್ಸ್ ದೇ ಕಂಡಕ್ಟೆಡ್. ಫಸ್ಟ್ ನೋಡಿ ಹೇಗೆ ಮಾಡ್ತಾರೆ. In one of the experiment, they grew some viruses on a medium having radioactive phosphorus. You know, radioactive phosphorus means P32. It's not normal phosphorus. You know, phosphorus is present only in DNA. Some others on medium having radioactive sulfur. Radioactive sulfur means S35. You know, sulfur is present in the protein but not in the DNA. Thus, he produced 2 types of virus. Virus grown in radioactive phosphorus have radioactive DNA. Carefully listen. Virus grown in radioactive phosphorus have radioactive DNA but not radioactive protein because phosphorus present in DNA not in protein. Second point, virus grown in radioactive sulfur medium have radioactive protein. Protein means protein coat, not the radioactive DNA. Because of this, sulfur present in protein but not in DNA. These two are very important point. So brief agandre DNA ನಲ್ಲಿ P32 ಇದೆ, ಪ್ರೋಟೀನ್ ನಲ್ಲಿ S32 ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಎಲಿಮೆಂಟ್ ಇದೆ. See the diagrams. ಅವನು ಇನ್ಫೆಕ್ಷನ್ ಮಾಡಿಸ್ತಾನೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಗೆ, ವೈರಸ್ನ ಇನ್ಫೆಕ್ಷನ್ ಮಾಡಿಸ್ತಾನೆ. ನಾವು ಮುಂಚೆ ಹೇಳಿದ ಥರ ವೈರಲ್ ಡಿ ಎನ್ ಎ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್ ಒಳಗಡೆ ಹೋಗುತ್ತೆ. ಹೋದ್ಮೇಲೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಡಿ ಎನ್ ಎ ಇದು ಕಂಟ್ರೋಲ್ ತಗೊಂಡು ವೈರಸ್ ಪಾರ್ಟಿಕಲ್ಸ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಗೊತ್ತಾಯ್ತಾ? ಇದೇ ಥರನೇ ಕೋವಿಡ್ನಲ್ಲೂ ಆಗೋದು ಸಹ. Now, radioactive phages were allowed to infect E.coli bacteria. The phages transfer the genetic material to the bacteria. The viral coats, how the viral coats are separated? The viral coats were separated from the bacterial surface with the help of a blender. Agitating blender ಅಂತ ಕರಿತೀವಿ. ವೈರಲ್ ಕೋಟ್ಸ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಗೋಸ್ಟ್ ಅಂತ ಕರಿತೀವಿ. ಖಾಲಿ ವೈರಲ್ ಕೋಟ್ ಇರುತ್ತಲ್ಲ ಅದನ್ನು, ಹೆಡ್. ದಿ ವೈರಸ್ ಪಾರ್ಟಿಕಲ್ಸ್ were separated. How the virus particles are separated from the bacteria? By centrifuge mechanism. When centrifugation done, 2 layers are formed depending upon the density. Now the observation. Bacteria infected with viruses that had radioactive DNA were radioactive. No radioactivity found in the supernatant. ಅಂದರೆ ಫಸ್ಟ್ ಎಕ್ಸ್ಪರಿಮೆಂಟು ಯಾವ ವೈರಸ್ನಲ್ಲಿ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಡಿ ಎನ್ ಎ ಇತ್ತೋ ಆ ವೈರಸ್ಗಳಲ್ಲಿ ಮಾತ್ರ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇದೆ. ಮೇಲೆ ಪ್ರೋಟೀನ್ ಸೂಪರ್ನೇಟೆಂಟ್ನಲ್ಲಿ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಇಲ್ಲ. ಎರಡನೇ ಅಬ್ಸರ್ವೇಷನ್ನು, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇನ್ಫೆಕ್ಟೆಡ್ ವಿತ್ ವೈರಸ್ ದಟ್ ಹ್ಯಾಡ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಪ್ರೋಟೀನ್ ವರ್ ನಾಟ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್. ಯಾವ ಪ್ರೋಟೀನ್ನಲ್ಲಿ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಇರ್ತಿತ್ತೋ ಇಲ್ಲಿ ರೇಡಿಯೋ ಆಕ್ಟಿವಿಟಿ ಸೂಪರ್ನೆಟೆಂಟ್ನಲ್ಲಿ ಮಾತ್ರ ಇರುತ್ತೆ, ಡಿ ಎನ್ ಎನಲ್ಲಿ ಇರೋದಿಲ್ಲ. ಕನ್ಕ್ಲೂಷನ್, ಡಿ ಎನ್ ಎ ಈಸ್ the genetic material. So by conducting Hershey and Chase experiment, they proved that transforming principle DNA, biochemical DNA, genetic material DNA, ಈ ಥರ ಸೈನ್ಸಲ್ಲಿ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ರಿಸರ್ಚ್ ನಡೀತಾ ಇರುತ್ತೆ, ಎಕ್ಸ್ಪರಿಮೆಂಟ್ಸ್ ಮಾಡಿ ಪ್ರೂವ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. ಸೊ ದಟ್ಸ್ ಆಲ್ ಅಬೌಟ್ ದಿಸ್ ಎಕ್ಸ್ಪರಿಮೆಂಟ್, ಹೆರೇಶಿ ಚೇಸ್ experiment for 5 marks. This experiment is for 5 marks. Now, we came to know that DNA acts as genetic material. There are chemicals like proteins, carbohydrates, DNA, RNA, etc. What are the criteria to behave as genetic material? It's important for 2 marks only. Criteria for genetic material. First point nodri. The genetic material ಆಗಬೇಕು ಅಂದರೆ it should be able to produce another copy of that one. That is, it should be able to replicate itself, duplicate itself. First criteria. Second criteria, it should be chemically and structurally stable. ನೋಡಿ, ಕೆಮಿಕಲ್ ಚೇಂಜಸ್ಸು ಆಗಬಾರ್ದು, ಸ್ಟ್ರಕ್ಚರಲ್ ಚೇಂಜಸ್ಸು ಆಗಬಾರ್ದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇನ್ ಗ್ರಿಫಿತ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹೀಟ್ ಮಾಡಿದಾಗ DNA ಕೆಮಿಕಲ್ ನೇಚರ್ ಏನು ಚೇಂಜ್ ಆಗಿಲ್ಲ. ಅದೇ ರೀತಿ ಸ್ಟ್ರಕ್ಚರ್ DNAದು ಚೇಂಜ್ ಆಗಿಲ್ಲ. ಇಟ್ ಮೀನ್ಸ್ ಕೆಮಿಕಲಿ ಆ್ಯಂಡ್ ಸ್ಟ್ರಕ್ಚರಲಿ ಸ್ಟೇಬಲ್. Another important point, it should provide slow changes. Slow changes means mutation necessary for evolution. ನಾವೀಗ ಹೇಳ್ತೀವಿ COVID-19ನಲ್ಲಿ ಮ್ಯೂಟೆಂಟ್ ವೆರೈಟಿ ಬಹಳಷ್ಟು ಇದೆ ಅಂತ. ಸೊ ನಮ್ಮ ಇಂಡಿಯಾದಲ್ಲೇ 85 ವೆರೈಟೀಸ್ ಇದೆ ಅಂತ ಹೇಳ್ತಾರೆ. ಅಮೇರಿಕಾದಲ್ಲಿ D614 ಈಸ್ ಹೈಲಿ ಡೇಂಜರಸ್ ಮ್ಯೂಟೆಂಟ್ ವೆರೈಟಿ ಅಂತ ಹೇಳ್ತಿದ್ದಾರೆ. ಅಂದರೆ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಆಗೋದು ಮ್ಯೂಟೇಷನ್ ಆಗಬೇಕಾದ್ರಲ್ಲಿ ಪಾಯಿಂಟ್. ಇಟ್ ಶುಡ್ ಬಿ ಏಬಲ್ ಟು ಮ್ಯೂಟೇಟ್. Another very important point is also there. It should be able to express itself in the form of Mendelian character. Mendelian character ನೀವು ಹೈಸ್ಕೂಲಲ್ಲಿ ಓದಿದರೆ ಗೊತ್ತಿರುತ್ತೆ. ಮೆಂಡಲ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟು, ಹೌ ದಿ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಆರ್ ಟ್ರಾನ್ಸ್ಮಿಟೆಡ್ ಫ್ರಮ್ ಪೇರೆಂಟ್ಸ್ ಟು ಆಫ್ಸ್ಪ್ರಿಂಗ್ ಅಂತ. ಸೊ ಡಾಮಿನೆಂಟು, ರೆಸೆಸಿವು ಆ ಥರ ಎಲ್ಲ ಇರೋದು. Protein does not fulfill the criteria, hence it is not the genetic material. RNA and DNA fulfill the criteria, hence RNA and DNA act as genetic material. Once again, see only 4 or 5 important points. It's for 5 marks. Thank you.